All right, ladies and gentlemen, welcome back. So we have sort of been alluding to classification schemes of prokaryotes really through this whole chapter. And up to this point, we've really been talking about the classification mechanisms that were utilized up until the late 20th century. Things from their shape to their ability to move to their nutritional modes, their response to gram staining. And to be clear, all of those things are still useful, um, but they really didn't provide a clear evolutionary history um, to where these prokaryotes came from, what their evolutionary connection was, who was more closely related, who was more distantly related. So, although the things we've been talking about thus far are still means by classifying and identifying these prokaryotic species, they failed to lack that clear evolutionary history. So once we started sequencing genomes, actually back in the 1970s, these guys were some of the very first DNA sequenced organisms because they're relatively simplistic. They don't have a lot of genes. They don't have a lot of DNA. Period. Plus, they're easy to culture and get a hold of, and so on and so forth. So they are the ones that we probably know the most about in terms of their genetics. What first came about of that sequencing in the 70s, though, was that we needed to split out those prokaryotes into two distinct domains. So pre all of the sequencing, and really even into the 90s, early 2000s, before this really took hold, there was a lot of question of needing to recognize the fact that we had kind of thrown all of these prokaryotes into one kingdom, and yet the more and more we dug into their sequencing, the more and more genetics we started studying and understanding, the more we realized that there were two distinct evolutionary tracks going on here. The eukaryotes all sort of stayed in their own domain, and then we split out into kingdoms, classes, phyla, so on and so forth. But when we looked at the prokaryotes, we realized we needed to split them into bacteria and archaea because those archaea species are actually more closely related to eukaryotes in terms of their genetics than the bacteria are. And so we needed to do some rearranging, which is where the three domain system came from. A couple other things we realized as we were working through molecular systematics on these prokaryotes is just the immense diversity that exists. They are one of the most genetically diverse categories of organisms on Earth. And back that train up a step, the fact that they literally can survive anywhere and everywhere probably makes sense when you think about it. They're going to need a huge diversity of genetics in order to deal with all of those different environments that they come across. The other thing that we started to realize, especially as we started trying to dig deeper into the roots of their tree, is that the significant horizontal gene transfer that exists between prokaryotic species, the fact that they can share plasmids, the fact that they can share genetic components, parts of their chromosome with each other between species, and they do it naturally on their own, really complicates these deep pieces back here, right? Really complicates the ability to start tracing back to that single common ancestor of life itself. Because those genomes really are these big mosaics, these big pieced together incorporations of a bunch of species when we look at their actual genomes themselves. So when we look across comparing those three domains and what components as a result of their genetics come out, here's what I mentioned a few moments ago, is that there's actually considerably more connection, both genetically, phylogenetically, even just characteristically, that connects those archaea more to the eukaryotes than it does to those bacteria, which is kind of interesting. It's not what you would think about, especially when you start digging into the fact that that archaea term kind of comes from ancient. And we'll see why that term made sense when they were initially looking at them as their own distinct group. So I'm just going to kind of throw a couple of examples out here. If you want to dig more into these major groups of bacteria and archaea, by all means go for it. There's some really amazing organisms out here, but I'm just going to throw out some of the classifications that are there. So once you get past that domain stage, there are classes or groups of organisms. They do sort of work through that class, phylum, genus kind of idea like we see in eukaryotes, but it's a little less straightforward. Um, they definitely have genus species names, so they will work that way, but then there can be strains within those species, and so it does get a little bit more complex. But just to kind of throw some pieces out there, proteobacters, these then all have alpha, beta, gamma, so Greek orientations to them. So these are just some examples here. Chlamydias are another group. Okay. Spirochetes get their own group. Gram-positive bacteria as a whole kind of get their own group, and then split out from there. And same with the cyanobacteria. What I do want to talk a few minutes about are the archaea, because these guys are really kind of interesting, especially given the fact that they are more closely related to eukaryotes, which when you start digging into their ecology and their life cycles, seems almost counterintuitive. So when we talk about archaea, you often may come across the term extremophiles because these are prokaryotes that really seem to live in some really extreme environments on Earth. And that term archaea came from the idea that we thought these were ancient because they live in places that would have been much more like early conditions on Earth. And yet it seems like those bacteria are actually older, which is kind of interesting. So they live in some really extreme environments. So these are some of the hydrothermal vents, hot springs. This is actually my picture from Yellowstone. This is Grand Prismatic, and it is a hot spring pool, and each of those different colors associates with a different archaeal species, a different bacterial species, prokaryotic species, excuse me, um, that is also connected to different temperatures. So the deeper blues here in the center are their own species that lives at a particular temperature. You get this green ring that is a slightly cooler temperature, the yellow, and then out into the sort of brownish burnt orange out here. And all of those are associated with different temperatures and as a result, different species that can live there, which is kind of cool. So thermophiles, these are a set of archaea that would live in really hot environments. So those hot springs, for example, or those thermal vents at the bottom of the ocean. And most of these species thrive somewhere around 60 to 80 degrees Celsius, or sometimes even hotter than that. Um, so Phyloris phylogeny, these guys are found down around the hot deep sea vents, and their temperatures can get upwards of 113 degrees Celsius, so considerably hotter. This is the Pyrococcus pyrorinus, and this one is where we get Taq polymerase from. We get the DNA polymerase source for PCR reactions, so being able to do DNA replication in a test tube outside of the cell. And we needed that DNA polymerase to be heat stabilized at a much higher temperature than human body cells or most organismal body cells natural temperatures because I need to denature the DNA. I need to pull the DNA apart in order to allow the DNA replication to happen, and we use heat to do it. So they needed a DNA polymerase to build those new DNA molecules that was stable at much higher temperatures in order to make that PCR work. This guy also found in a hot spring in Yellowstone, which is pretty cool. Um, Halophiles, these guys live in extremely salty environments, so places like the Dead Sea, Great Salt Lake. Those would be some examples of where you'd find halophiles. Methanogens, these are another big one, and we use a lot of these guys for industrial purposes, like they're incredibly good decomposers. And so we use them in sewage treatment facilities as a way to break down larger waste products and be able to clean the water that we get from them. So that's pretty cool. They also naturally occur in a lot of swamps and marshes, so a lot of anoxic environments. They are strict anaerobes, and they also exist in a lot of guts of cattle. So they have formed symbiotic relationships by helping break down, I should say, a lot of materials, a lot of times cellulosic materials. So coming out of things like the grasses and the plants and things that they're eating. Um, but then they produce methane as a waste product. So uh, they tend to be a little stinky as a result of that.